Hi, thanks for coming along to this Darwin in Shrewsbury Festival lecture about evolution happening before our eyes, how viruses evolve. So we have Dr. Daniel Goldhill here from Imperial College London, Public Health England, who's here to give a really interesting lecture. So Dr. Daniel Goldhill did his undergraduate degree in biology at Oxford, where he was inspired by reading about evolution, but really wanted to do experiments to make it happen. He did his PhD in ecology and evolutionary biology at Yale University, evolving viruses of bacteria in the lab to try to understand how mutations in the genetic material led to changes in proteins and properties of the virus. After his PhD, he moved to the UK to do a joint position between Public Health England and Imperial College London to work on the influenza virus. His project included evolving influenza virus in the lab to become resistant to new antiviral drugs that could be used against the flu. In 2020, he's been part of the effort to understand COVID-19 and as a member of Professor Wendy Barclay's lab at Imperial College. Though he hasn't been evolving the virus in the lab yet, he has been trying to understand what are the changes to the virus that make COVID-19 able to cause a pandemic. So really exciting lecture. Just to let everyone know, we are recording this and we're hoping that we can make it available online. If you've got any questions throughout, you can use the Q&A button at the bottom to ask, but we'll take all of those at the end. Right, Dr. Goldhill, over to you.
Thank you so much. Thank you everyone for coming. I'm an evolutionary biologist and I study how viruses evolve, and I try to evolve those viruses in the lab. And I've had a very busy year. And I'm going to be talking to you today about two viruses mostly about influenza virus and then about SARS-CoV-2, the virus which caused the current pandemic. So I thought I'd start with Darwin because this is the Darwin Festival. And Darwin studied finches, which you would know if you attended the talk yesterday, but he also studied pigeons, and he bred pigeons in his house. And he found that he could select various different types which had various different characteristics, which were all quite different. And he felt that this was a really good example of selection happening on the birds, and a really good example that led him to develop the theory of evolution. Now, Darwin obviously didn't know about viruses. Viruses weren't really understood until later. But if he had known of them, I think he would have really liked them, because viruses are one of the easiest things to demonstrate evolution with in the lab. So let's talk about what a virus is. Viruses are parasites. They can only replicate inside another cell, and they have very small genomes. And the genome is the information that codes for the proteins that make up the virus. And this genome can be made out of DNA, like you have, or it can be an RNA genome, which is a slightly different type of genome. And it's contained inside a capsid, so a sort of a shell around the genome. And there's not much biology of viruses that you need to know for this talk, but you should know that lots of viruses have spike proteins on the outside. And these spike proteins are used to attach to and enter the cell. And viruses carry about with them their polymerase, which is an enzyme which they can use to copy their own genome. So they use lots of material from the host, but they also have some of their own proteins which they can use to make more copies of themselves. Now, some people debate whether a virus is alive or not. And that's not something that's an interesting question to me. And the reason it's not an interesting question to me is because we know that viruses can definitely evolve. And what I'm really interested in is evolution of viruses. So the fact that they can evolve and change means that we can study them. And viruses evolve really fast. They make lots of errors when copying their genomes. So in my own research, I've found that I can evolve resistance to some drugs in the lab in a week. And we can see worldwide that you can have new variants of a virus that can spread worldwide in under a year and completely wipe out all other viruses of that species. And one really nice thing about working with viruses in the lab, which I'm going to talk a bit about later, is that you can store viruses in the freezer and you can get them out again. So I can, on any given day, compare a flu virus from the 1960s, the 1990s, and from 2010. And I can see how the viruses change. So let's talk about pandemics. How do they start? Well, I first heard about the COVID-19 pandemic in an email that was talked about from a particular mailing list, which looks to find various disease outbreaks. And this was sent out right at the end of 2019. It said there was an undiagnosed pneumonia in China, and they wanted to know if anyone knew anything more about it. And scientists saw this, and lots of them started to worry, and we know where it ended up. But more generally, what are the steps that a virus needs to take to get to a pandemic? So step one is the virus has to circulate in animals. Most pandemics are viruses which jump from animals into humans. And because humans haven't seen these viruses before, they can spread throughout the population. So the first step is that the virus has to circulate in animals. The second step is that it has to jump from an animal into a human, a process that we call viral emergence. The third step is that we then have to have human-to-human transmission. And if that human-to-human transmission is successful, then we can have a pandemic, global spread of the virus. And after the pandemic, the virus will continue to evolve. The virus could adapt further to humans, and it could become an endemic virus, a virus that's always present, or a seasonal virus, a virus that comes back like flu each year. Now, I've drawn this diagram on the right to show the different, each of the different steps gets smaller and smaller. So there are lots and lots of viruses that circulate in animals, and only a handful of those will jump into humans, and only a handful of those will have human-to-human transmission, and only some of those will cause a pandemic. So let's look in the steps at slightly more detail. So how many viruses are there out there? It's estimated that there are 1.7 million undiscovered viruses in mammals and birds. And you can see some of the culprits of that have led to various pandemics in the past, shown below. And the sheer diversity of viruses means that it is impossible to discover every single virus in mammals and birds. We have only seen a fraction of what is out there. So we don't know what viruses still remain out there that could potentially cause a pandemic. So the next step is the virus has to emerge. It has to jump from animals to humans. And this can happen in a lot of different ways. Now, and there are lots of ways which modern humans have, which potentially might not happen for other species, for example. So one of those is zoos. There are some viruses which are found in animals, which when we test zookeepers, we can find that the zookeepers have antibodies to those viruses, but they very rarely spread them on to other humans. But it's one particular way where humans come into contact with animals that can spread viruses. So another way that it can happen is through farming. So having large numbers of animals very in close proximity is a really good way to spread disease. And those diseases can jump from animals into humans. So many viruses are thought to have jumped into humans once humans developed agriculture and started farming cows, for example. Another way that that viruses can jump from animals to humans is through the process of hunting wild animals. And this is often thought at how Ebola outbreaks start, for example, that communities might hunt other primates, and the process of hunting is a really bloody process, and there might be small cuts on the hunter's hands, and blood from the animal can get into the hunter like that. Or alternatively, people will find a dead animal in the forest and they will bring it back and eat it, and outbreaks can potentially start like that. And it's also worth saying that that there are cultural differences which can lead to greater likelihood of outbreaks. So if you're in a particular culture which has live poultry markets, then that's a lot more people who are coming into contact with animals than in a culture where your birds are wrapped up in the supermarket. And the final way that I'd like to mention that you can potentially have an outbreak is through pets. It's another way that humans come into contact with animals. So lots of different ways that viruses can jump from animals into humans. But just because a virus jumps from an animal into a human doesn't mean that it will jump from a human into a human. So human-to-human transmission, it could happen through respiratory droplets. It can happen depending on the virus. It could happen through sexual contact. It could happen through sharing of bodily fluids, if you share needles, for example. Viruses which have come from animals aren't guaranteed. Just because they can infect one human doesn't mean that they'll be able to go and infect others. So a really good example of this is rabies. Lots and lots of humans have had rabies, but most humans don't go on to transmit rabies to other humans, and there isn't a worldwide pandemic of rabid humans. So after human-to-human transmission, if it occurs, then we have a pandemic, global spread. And currently, we're living in a golden era, really, for the potential of pandemics due to the huge increase of global travel. So you can be in any country in the world and travel to all 6 continents very within 24 hours, and viruses can therefore spread throughout the world in a much faster way than ever before. And it's also worth saying that at the moment we're coming into contact with more wild animals through habitat destruction. And it's forcing some humans and animals to live closer together. And that also is increasing the risk of an animal to human jump, human to human spread, and then global spread in a pandemic. And once the virus is spreading exponentially, you get exponential growth. And as we know, the virus can be very difficult to stop. Now, it's worth saying that not all viruses that have demonstrated the ability to spread between humans go on to cause a pandemic. So just as not all viruses that can cause an infection in one human can spread to humans, other humans, not all viruses which can be spread from humans will cause a pandemic. So for example, bird flu has caused very small epidemics with limited person-to-person spread, but it's never been able to go pandemic. It doesn't have exponential growth. Each person isn't going to be infecting multiple other people, and so on and so forth. So the animation that you're seeing is from a site called nextstrain.org, which I would definitely recommend that you check out, as it's a fantastic resource for learning about evolution of viruses. And it's showing the spread of the SARS-CoV-2 virus causing COVID-19 from China. And you can see how global travel has led viruses to spread throughout the world and go from continent to continent in both directions. So even if you have a pandemic, it's not necessarily going to be a deadly virus, though those are obviously the ones that we care about. So what makes a virus deadly? And I'm now going to talk you through an example of a previous pandemic and look at how we've researched what made that virus deadly. So it's been about a century since the 1919 flu pandemic, the first really big flu pandemic. And in this pandemic, it was estimated that about 40 to 100 million people died worldwide. And there's been lots of similarities seen between the 1918-19 flu pandemic and the current pandemic that we're living through. And in fact, I thought I'd share this picture. I used to play football in this gym, and you can see that just like in 1919, it was repurposed and laid out with beds in case there was a huge increase in COVID patients. So 1918-19 flu differs from coronavirus, however, in some really crucial ways. So, for example, flu in general has quite high mortality in the very young. And so it was not surprising to see that there was an increase, there was a high death rate in young people, in the very young. But also 1918-19 flu had this very odd W shape where there was a peak in mortality in young people, aged sort of 25 to 34. You can imagine that if we'd seen a curve that had looked like this with large numbers of deaths in young people and in very small babies, we might have had we would have had an even more deadly pandemic than SARS-CoV-2. And we do have some understanding of what caused this high mortality in young people, and that there are a few possibilities, but the one that we think is most important is that it caused what's called a cytokine storm. And you don't need to know exactly what that means, but it's the body's own immune system is fighting the virus so hard that it ends up killing you. And in fact, a cytokine storm is of a different sort, is also what is thought to make SARS-CoV-2 so deadly. So if we want to understand what about the 1918 flu virus made it so deadly compared to, say, current flu viruses, what we'd really like to do is have a virus sample from 1919. But how do we do that? People weren't saving viruses in their freezers back then. So one scientist realized that bodies buried in the permafrost could act as a natural freezer. And so he traveled in the 1950s to a place called Brevig Mission in Alaska. And if anyone felt that the weather was cold, I looked up the weather in Brevig Mission today, and you can see that the high was minus 26 degrees. So really cold. And he went in the 1950s, and with permission of the community, he excavated some bodies, and he tried to isolate the virus, but he was unable to. Now, fast forward 40 years, and scientists had new techniques. They could now sequence the virus. And not only could they sequence the virus, we now have the tools to recreate the virus in the lab solely from the sequence itself. So solely from the genetic code of the RNA in flu's case, we can sequence that RNA. And once we have the code that codes for the virus, we can then make the virus from scratch in the lab. So the scientists returned to Breivik mission and they re-excavated a body. And this time they weren't looking for intact virus. They were trying to see if any of those virus genomes were still could still be found. And amazingly, they were successful. They managed to extract enough information to completely recreate a virus from the beginning stage of the pandemic. And now, because we've recreated that virus in the lab, we can compare it to current viruses, and we can mix and match different parts of the virus, and we can look at individual mutations, and we can identify which bits of the virus were so important at causing serious disease. And there you can see Terence Tumpey, one of the scientists who was involved in the effort. Now, after a pandemic, the virus can adapt to humans. So we don't know exactly what the fatality rate was for the pandemic flu virus in 1918, 1919, around 1.5–2%. But what we do know is that the virus continued to circulate, and it still circulates today. Some segments of that virus are still in influenza today, but obviously it has much lower mortality. And one of the ways that it's done that is it's adapted to humans, and it no longer antagonizes the human immune system to such a large degree. Now, it's worth saying that when a virus jumps into humans, and when it emerges in humans, or in any other species, we can't predict whether the virus will become more virulent or less virulent. So if you see anyone saying viruses have to become less virulent, so SARS-CoV-2 is going to become less virulent in a few years, we don't know. So don't believe them. Now, flu is a really interesting virus, and we have a vaccine against 4 different types of flu, and this is one type of flu shown here called H3N2, and you can see this is a tree, a phylogenetic tree, and you can think of it as being a bit like a family tree for the virus. And different virus samples as you move through time, and as the samples move up, you can see that it's the number of different mutations, so they're gradually changing over time. And this is a process called antigenic drift, the accumulation of mutations over time. And in flu, what this means is that each year we have to look at the virus and decide whether we need a new vaccine. Has the virus changed so much that we need a new vaccine? So vaccines will create antibodies, and antibodies will bind to the spike protein for flu virus, which in this case is called hemagglutinin. And if the virus mutates its hemagglutinin at the sites where antibodies bind, it can throw off immunity, and that can occasionally mean that it will escape the immunity made by vaccines. Now, the vaccine strain has to be chosen 6 months before the flu season to give enough time to make enough vaccine for everyone to have some. So here is what the situation looked like in 2019. You have to choose which of these different viruses is going to be the virus that dominates. And it's very difficult. There are lots of different types. Lots of them are similar, but some of them are different, are antigenically different. So they have different, they look slightly different to the antibodies. And if they're antigenically different enough, it will throw off the antibodies, the vaccine won't work, and a lot more people will die in the upcoming flu season. So here you could try and, you might think that you should pick your vaccine from the top one. You might think it should maybe be the bottom one. It's difficult to know which one is going to spread the most. And in fact, you can see you might have picked from here and you would have been wrong, because these viruses no longer circulated, and in fact it was other clades which ended up being most successful. Now, flu is an example of a virus that changes and we have to update the vaccine for. There are some viruses where you can have a vaccine or you get infected as a child and you're protected for life. And trying to understand the differences, why can one virus evolve away from this immunity and you can get reinfected lots of times, and why can some viruses not evolve away from the immunity? There's constraint on the virus is a really interesting question for evolutionary virologists. So I'm now going to move on and talk about coronavirus and coronaviruses. And here are some pictures taken with an electron microscope of virus. And this is virus that I grew a few weeks ago, taken from our collaborator. And you can see that you can faintly see the spike of the coronavirus, which gave it a sort of, which some people thought looked like a crown, which is why it's called coronavirus. And here you can see some of the viruses coming out of a cell. So what is SARS-CoV-2? It's the virus that causes COVID-19. It's a coronavirus. And you remember that I said that viruses have very small genomes. So coronaviruses are actually the biggest genomes for RNA viruses. They have 30,000 nucleotides. You have 3 billion nucleotides. So if you printed them out, which someone did, it would be about 130 volumes. And coronavirus would be less than a single page in terms of size differences. So what do we know about coronaviruses? So in 2000, if you'd asked a scientist what are coronaviruses, they would have said they cause the common cold. There are 4 different coronaviruses which we know of, which are some of the viruses that cause common cold. So don't cause serious disease in humans, and they cause some slightly more serious diseases in animals, so in cats or in pigs. Now, in 2003, there was the first epidemic of coronavirus, and this was SARS, or SARS-CoV-1, as it's now known. And this was a virus that went from bats to another, to an intermediate host, which we don't know exactly which one. And then it led to headlines like this. And it led to an epidemic, which caused lockdowns in Hong Kong, and it caused a lockdown in Toronto in Canada. But it was contained, and it only killed around 700, 800 people. And then in 2005, another coronavirus emerged, this time probably originally from a bat, but this time it spread to humans from camels. And this is particularly pathogenic, and that out of the known cases where people have been infected with MERS, 34% of them have died. So that leaves us with a graph that looks a bit like this. We've got the common cold viruses down here. They're transmissible. We know that. They're very transmissible. They don't kill anybody. And then we have MERS and SARS, which are over here. We know that they have a much higher mortality rate, but we know that they're not particularly transmissible because they didn't cause a worldwide pandemic. So really, that should have led us to the question, can coronaviruses be here with a high mortality rate and very transmissible? Possibly not. There might be trade-offs between those two things. But could we have coronaviruses here, which cause a little bit of mortality, but they're still pretty transmissible? That might be the recipe for a pandemic. And of course, we know that's what happened with SARS-CoV-2. So where did SARS-CoV-2 come from? It's closely related to SARS-CoV-1, but its closest relative is in bats. And we don't know whether it came to us through an intermediate host, like a mammal, probably through like SARS-CoV-1, or even something more exotic, like a pangolin. We're not sure. And one of the reasons we're not sure is partly because one of the reasons why we couldn't see this virus coming. And that's because there are lots of coronaviruses circulating in bats, and these viruses can recombine. And what I mean by this is when two viruses both infect the same bat, they co-infect the same bat, they can trade bits of their genome. And this causes a huge amount of viral diversity. So for example, SARS-CoV-2, one of the reasons why we think potentially pangolins might be involved is there's a key bit of the spike protein of SARS-CoV-2, which looks most closely related to a virus found in pangolins, even if the rest of the virus looks like it came from bats. So very possibly, this is an example of recombination in bats. So why can't we just go out and test all the bats? Well, bats are a bit funny, and that even though they have a lot of viruses, for various reasons, they're very good at keeping levels of virus low. So it's very difficult to go out and find live viruses in bats. But we know through antibody testing, for example, that bats have had these viruses. So SARS coronavirus and SARS coronavirus 2, even though they were quite similar, had wildly different outcomes. So I put a little timeline here going from the first case of SARS coronavirus versus SARS coronavirus 2. You can see WHO was informed much quicker for SARS coronavirus 2. And after just around 80 days, SARS coronavirus had overtaken the total, SARS coronavirus 2 had overtaken the total number of cases and deaths for SARS coronavirus. And then it went on spreading exponentially to cause millions more cases and over 1 million deaths now. So can we compare SARS coronavirus 2 to other viruses to try to work out why it caused the pandemic? So yes, we can do this. And one of the things that we've been looking at in my lab is this particular insertion into a cleavage site in spike. So what the insertion does isn't that important, but you can see that in SARS coronavirus 2, it has these 4 extra amino acids, so the building blocks of proteins, amino acids. SARS coronavirus 2 has 4 extra ones in a key place, which makes it more easily cut at that point. And the most closely related viruses in bats and pangolins don't have those amino acids, nor does SARS coronavirus 1 have those. And what we've done in lab is we found SARS coronavirus 2 viruses with and without these 4 amino acids. And we can show that having those 4 amino acids leads to better virus growth in respiratory cells and better transmission. So this might not be the only reason why SARS coronavirus 2 caused the pandemic, but it appears to be a key reason why it appears to be essential for SARS coronavirus 2. And we do see these cleavage sites in other coronaviruses, not the most closely related ones. So again, there's a question, is there some sort of recombination event that happened? So how is SARS coronavirus 2 evolving now? So SARS coronavirus 2 throughout the pandemic has had about 1 or 2 mutations in its genome on average a month. And most of these mutations will make no difference to the virus, and they will definitely not make any difference to us. However, as I'm sure you're aware, that's not the case with every mutation. So, sorry, this is again from NextStrain, just showing the diversity of the different viruses that are out there. Now, each one sort of often evolving, there's a lot of diversity across all the different continents. So one mutation that has ended up proving to be important is this mutation called D614G. And when I give you a number, it just refers to the particular amino acid number in the spike protein. It's not too important. But what was seen worldwide is that the G variant happened to dominate as we went through 2020. But it's really difficult to understand the importance of this, because mutations that you see at the beginning of the outbreak, it might just be chance. So we saw that G614 emerged in Europe, and we know that Europe was driving the worldwide outbreak in February and March. However, this particular mutation, we have gone on to do the associated lab experiments, and we can now show that it does help, it does change the virus spike protein and give the virus a bit of an advantage, even if it doesn't really change the disease course for humans. So how is SARS coronavirus evolving in the UK now? Well, the answer is there are new variants. And in particular, there's this variant called B.1.1.7, which has lots and lots of mutations. So this particular, there are also other variants, B.1.351, a variant which was first discovered in South Africa, and a variant which was first discovered in Brazil. And I'm going to talk mostly about the UK variant, B.117. So it probably arose somewhere in Kent or East London. And you can see in this animation how it spread from that part of England throughout the rest of the UK to dominate as we went through December. And shown in pink here, you can see again a slightly different way of looking at it. This virus has suddenly dominated. So what's going on with these variants? Where have they come from? Why are they suddenly so successful? So these variants have lots of different mutations in the spike protein, and they share some of these mutations. And when you see parallel evolution like this in different lineages, which are all rising at the same time, it's likely to be a sign of positive selection. And there's some evidence that these particular mutations working together do seem to lead to tighter binding to the spike protein, which I'll get onto in just a little bit. So where did the UK variant come from? So one possibility is that it came from an immunocompromised person, someone who might have the virus for an extended period of time without being able to clear it, because their immune system isn't able to get rid of the virus, but they're not dying. The virus is just replicating and evolving in them, and then potentially spreading to other people. So there's a couple of reasons why we think an immunocompromised person could be responsible. The first is that there were lots of mutations in a short period of time without there being any intermediates. So this shows again a sort of family tree of the viruses, And we can see once the lineage B.1.1.7 is first sequences, there's lots and lots of diversity. So it's spreading really rapidly, but there's no intermediates suggesting that it was suggesting that it went through one person to another and it sort of got the mutations in sequence. It sort of came fully formed with all the mutations. And that suggests that they could have all evolved within one person. And in addition, the mutations that were seen in the UK variant were also shared with mutations that have been seen in immunocompromised patients. So that's one possibility where it came from. And as I was saying before, what do these mutations do? Some of these mutations are in the spike protein, and we know that they're right where they bind to the receptor. So they might be affecting how tightly the spike protein binds to the receptor. But this may also reduce antibody effectiveness. So the antibodies that your body makes in response to a vaccine or in response to infection, they really like to bind the top of the spike protein. So having these mutations might throw off some of those antibodies, possibly. I think we don't yet know the effect that the variants are going to have on all the vaccines. We definitely know there's some vaccines which still are very effective against current variants, and some which appear to be slightly less so. But potentially even, I think it's still an open question. So one additional mutation that's in the UK variant changes the insertion that's in the cleavage site. It changes it from what was previously a proline to a histidine. And my lab is now looking to see, is that change important for the virus? So what's next? I think it's really important to say that we don't know why these variants are more successful at the moment. There could be lots of reasons. It could be that they're growing better. It could be that they're evading immunity. It could be that they're surviving longer in the environment. They're better at getting into the first cell. They're better at transmitting for some reason. We don't know. But what's clear is that there are probably going to be more variants. And these variants might change their antigenicity, and it might require updated vaccines. But one really exciting thing is that there's a whole batch of vaccines which are made directly by copying the virus's genetic code. And those vaccines will be relatively easy to update. So hopefully we'll be able to stay ahead of the virus. So I've put a couple of links of interest up. Nextstrain.org is really great, and the same people behind that have made covariants.org if you're interested in learning about the variants of the coronavirus. I'd like to acknowledge my lab, who helped a lot with this, and I'm really happy to take any questions. So.
Hi, Daniel. Thanks very much for that. I think we've got one question in the Q&A so far, which is from Helen. The question is, are there any plant viruses and are they transmissible to humans, and are any dangerous?
There are definitely plant viruses. They're viruses of pretty much everything has viruses. There are even some viruses which have sort of viruses of their own. I don't think that, I can't think of any examples of plant viruses that have jumped to humans. And in general, when we're thinking about viruses that can jump to humans, we expect the virus is going to be most likely to jump to a human if it's in something that's most closely related to humans. So if there's a virus that could infect a chimpanzee, then we'd be worried. And and if it could infect a mammal, we'd be more worried than if it infected a fish. So again, I don't think there are viruses of fish which infect humans. So our main, so there are definitely viruses which infect plants, and they can cause a lot of economic damage. Do you want to pick another question, Helen?
Yeah, if anyone would like to ask a question, if you could just put those into the Q&A box, and we'll pick some out of there. So we've got another question here. Do we know why some patients are so badly affected and others not from the same virus?
That's an excellent question. We don't really know that at the moment. We know there's some things that make people more likely to be badly affected. So age being an obvious one. And for some proportion of patients, there have been studies done where they have particular changes to particular genetic changes, which mean that they're going to respond poorly to the virus. So if you look up autoantibodies, that's one it gets quite complicated. If you look up, there's been papers where that explain about 10%, I think, 10 to 15% of people who get seriously ill with SARS coronavirus 2, they've got something wrong with an immune system gene. So they're missing something, they're missing an immune system gene, or they've made antibodies which stop their immune system working. And the virus can then grow better and the patients seem to do more poorly. But that, that's only explaining a small proportion. Other than that, it's really difficult to point to why some people do well and some people do poorly. And there are examples where they're twins, where one has done fine and the other hasn't, and we don't know. We're trying to figure out. We don't know. Lots of questions I can see.
Brilliant, thank you. I've just got another one here from Paul. Do you think it's possible to completely wipe out COVID-19 like they did with smallpox, or will coronavirus always find a way to mutate?
It's a great question. It's going to be very difficult. Smallpox is a fantastic example because that's a virus that that's pretty much one of the only viruses to have been eradicated. So something like polio is still hanging on by its fingernails in like a couple of countries in the world where it's difficult to do immunization programs. So we don't know enough about SARS coronavirus 2 and how it's going to respond, how it's going to change. With any virus, the fewer infections that there are, the less chance the virus has to change. So one of the reasons why we have lots and lots of variants is that there were lots and lots of infections, and that makes the chance of a rare occurrence that much more likely. We've never tried to wipe out a virus like coronavirus. I don't know if it's possible, but I wouldn't have necessarily thought that you could lock down a whole country and beat the virus like that. So I think we all have lots to learn about this. So sorry, I can't say one way or another. Like it might be possible, but I think it would be difficult.
That's great, thank you. I've got a question here from Chris. Will giving the Pfizer vaccine with a 12-week interval risk the virus developing resistance to the vaccine, similar to the immunocompromised person?
Really interesting question. At the moment, I would say that there are a lot more people out there who are getting the virus who haven't been vaccinated than people who might be getting the virus having had the vaccine. So the how the virus, the actual number of people who've been vaccinated probably aren't really going to be affecting the virus very much at this moment. So with flu, for example, we sort of expect that we don't really vaccinate enough people to put selective pressure on flu. I have seen people worry about it, that partial immunity could potentially lead to immune escape. I think the evidence for it is pretty weak at the moment, because we don't really know how the virus is responding to immunity, and that you're less likely to get infected even with the vaccine anyway. So there are fewer chances for the virus to mutate. And at this, yeah. So the other thing that I would add, that at this stage, the epidemiologist worked out that it was much better to vaccinate more people. That would do more to slow down the spread of the virus, which is probably going to have more of a difference which is going to make more of a difference to the virus evolving than patients who've had a slightly longer wait. So out of everything to worry about, I wouldn't put that in the top.
Brilliant, thank you. We've had a couple of questions about this. Just following up on the 1919 epidemic, and why there was a higher death rate within the younger age group of 25 to 35-year-olds. Do we know what happened with this group?
So that's So it's a really weird. It's interesting. So one of it is that you've got a really healthy immune system, and people's immune systems do change over time. So apparently at that point you would have a very healthy immune system, so you can make the biggest reaction and to the virus, but that reaction is itself what kills you. So that's one possibility, that those people just made a really massive reaction to it. There are also people who are out there and catching it as well. There is another possibility, which is that some people might potentially have been protected from, if they'd had flu in a previous epidemic. That's a lot. That we know a lot less about that, because the data just isn't there. It's a bit guessy. But the one possibility is that other groups might have had some protection, and if you get infected with the wrong subtype, it can lead to more problems. My best guess is that it's just that they had really healthy immune systems and they had high cytokine storm, and that killed them.
Brilliant, thank you. We've got a question here from Frank. What attracted you to study biology and then to specialise in viruses?
Thanks. So honestly, the thing that attracted me most was that I was really interested in evolution, and when I was a student, I read about evolution, and I did computer models of evolution, and I did some sort of computer models with evolutionary games, trying to understand Prisoner's Dilemma, for example, and how competition between different animals. And what I found is that it was cool to be able to do it on a computer, but it was even cooler to be able to do it in a lab. And if you wanted to do it in a lab with real things, you had to choose either bacteria or viruses. And I happened to pick viruses. And it's been really lucky because viruses have really small genomes. And so if you want to understand exactly it's much easier to try to understand what an individual change does. It's still really difficult to understand what an individual change does. So, you know, with the new UK variant, it's got 17 mutations that make changes to the virus. And these mutations, we don't know which are the important ones, and we don't know if they're interacting together. And it's going to take years potentially to piece together exactly how it's becoming more transmissible. But you can do that in a virus, and it's really difficult to do in a big animal, which is a lot more complicated. So if you want to get into the nitty-gritty of evolution, then I totally recommend studying viruses. And it's also been nice to be able to study an organism that makes a difference to people as well. So I've done some stuff with like looking at drugs and viruses, and those drugs could potentially be used, and it's nice to know that at some point down the line your research might make a difference.
Oh, that's brilliant, thank you. When growing cultures of viruses in a lab, how extensive are measures put in place to stop anything getting out?
That's a great question. So there are 4 sort of levels of safety, and they're called sort of CL1, CL2, CL3, and CL4. And CL4 is the most secure, and that's where we'd grow viruses such as Ebola virus, for example, which are, you know, going to be really, really dangerous to you. We do now have a vaccine, but that's only recent, and could potentially cause, you know, real problems. So SARS coronavirus 2, we grow at CL3, which is the level just below that. And the virus is always inside what's called a hood. So this is something with carefully controlled airflow, which then goes through a filter, which catches, which prevents any viral particles from going out a HEPA filter. And that, so we only work with the viruses inside the hood, but there are lots of layers on top of that. So for example, that hood is in itself contained within a negative pressure room, so that no air can escape from that room. And of course, we also have to wear appropriate PPE. So we're always wearing, when we're working with a virus, we're wearing 2 pairs of gloves, and we always take off the outer pair of gloves before exiting the hood. So there are layers upon, there are layers on top of layers of safety. So that's so that would be CL3. So there are more dangerous viruses which you take greater precautions for, where you have to sort of shower in and out of the lab every time you go in. The way it works when I work with the virus is that I keep the virus isolated. So I try and make sure that the virus can't get out. You can also work with the virus where you're in a sort of suit, and you have to do that if you're working with animals, for example, where you're in an airtight suit. So lots of safety precautions.
It's reassuring. Thanks. Are virus mutations totally random, or is there some pattern to them?
That is a really big question. So in general, in general, the mutations that happen to the virus are going to mostly appear random. But that's what's happening to the virus. The mutations we see definitely won't appear random, because if a mutation is deleterious, so it's really bad for the virus because it messes up a virus protein somehow, we're not going to see viruses with those mutations. So we're not going to see those mutations. So if we're thinking about the variants, then some of the mutations might just be random ones that the virus doesn't mind whether it has this particular bit or this particular amino acid or that amino acid. So there was a mutation there, it doesn't matter. We're going to see the mutations, we're going to mostly see, but then we're also going to see some mutations which are repeated, and those mutations might be more important. there's complicated biology behind whether the mutations are truly random. There might be particular reasons why some particular bases are more likely to mutate, and some particular mutations are slightly more likely to happen than others. But that doesn't really make much difference. Like the polymerase, the thing that copies the virus genome, just goes along. Every so often it makes a mistake, and it's pretty random where those mistakes happen for the most part. But the ones that we see won't be random.
That's fantastic, thank you. Right, I'll just select one more because I'm aware it's 8:30, although I think we could just keep asking you questions forever. I've just got one here, hopefully round us off, just saying, are there any helpful viruses that affect our bodies like those helpful bacteria?
Yes, there helpful viruses. So yes, there are helpful viruses. So we do have viruses within us as well, which are mostly viruses of bacteria, which we call bacteriophage. One really cool thing that my old lab is doing is they're looking at using bacteriophage, so viruses of bacteria, to try to treat antibiotic resistant infections. So those viruses could be really helpful. And what's really cool about these viruses is that these viruses can get in through, for example, these viruses can get in through a thing that pumps the antibiotic out. So if the bacteria wants to get around the virus infecting it by getting rid of its entry of how the virus gets in, it might resensitize it to antibiotics. In a really nice trade-off. So those viruses are definitely helpful. I think they're probably viruses within our gut ecosystem that are helpful as well. I know less about that, sorry. But
no, that's brilliant, thank you very much. And a massive thank you for this talk. It's been extremely interesting, very of the time as well. So yeah, it's really great to get that sort of background knowledge of what's going on and explained to us in such an accessible way. So thank you so much, Dr. Goldhill. And thank you to everyone that's come along and listened. We hope you're able to come to some more of the Darwin Festival lectures that we've got going on this week. Right, okay, thank you very much.
Thank you so much for listening. Thank you.